Hello, and welcome to the second Sunday Speaker Series for January. Today we have a very special speaker, a good friend of mine, and he is going to present on the topic of diatoms. What are diatoms, you ask? Well, a very important part of our ecosystem, and you're going to find out right now what importance it has and how it relates to Hartman Reserve. My friend, Dr. Lynn Brandt, is a UNI Emeritus Professor of Geology, and he'll tell you a little bit more about himself in just a second here. So, Lynn?
Greetings, everyone. I'm Lynn Brandt, and I taught geology at UNI for 25 years, but I also have some background in biology. And over the years, I got more involved in the study of diatoms. And so I want to talk a little bit about diatoms here in general and at Hartman Reserve. When we walk around Hartman Reserve, we see only a small part of what is here and what is going on in this place. We usually don't see what happens here at night, or what goes on underground, or what is acting too slowly to be noticed. We also miss a lot of things that are too small to see without resorting to a microscope. All those bacteria, plant spores, micro insects. Then there are diatoms. What I want to talk about today are these tiny organisms that play an extremely important role in the life of the planet, including your life, and which is also live in Hartman. Life on this planet is dependent upon the sun. Organisms of many kinds capture sunlight in the process we call photosynthesis. These organisms use the energy of the sun to produce sugars, starches, oils, and other molecules to make up their bodies. These organisms are at the bottom of the food chain. The photosynthesis here at Hartman is mostly accomplished by trees and lower growing green plants. The trees and other green plants capture sun's energy to produce food for all those who cannot make their own food. Deer, for instance, must rely on plants to live. Even those animals that eat other animals depend on some animal to eat plants. We might eat a hamburger, but that cow we are eating had to eat grass or other plants that captured the sun's energy. Eagles eat fish that might have eaten smaller fish that ate insects that ate diatoms. This is called a food chain. Plants and other things that photosynthesize also produce oxygen in that process. Without organisms photosynthesizing, our atmosphere would be devoid of oxygen. In aquatic environments, diatoms are a major part of the base of that food chain, and diatoms produce a large part of the oxygen we breathe. It is estimated that the oxygen in one breath out of every 4 or 5 that we take is produced by diatoms, mostly in the oceans. We also have diatoms here at Hartman, and I want to talk a bit about diatoms and show you pictures of some found here. Diatoms are small. Here I'm holding a microscope slide with diatoms mounted under a cover slip of 1 inch by 1 inch. There can be hundreds of thousands to more than 1 million diatoms on each slide, such as this. So what are diatoms? These are a group of algae that are single-celled, and they produce a cell wall out of silica. Essentially, these organisms live in glass houses. The glass is made by them out of silica, dissolved in the water in which they live. Waters that contain no silica, and these are rare indeed, do not have diatoms. But almost all natural waters except rain do contain silica. The cell walls of diatoms come in a variety of shapes and are intricate in their details. In fact, some of their smaller features are too small to see with a standard light microscope. The identification and classification of diatoms is based almost entirely on the shape and structures of the cell wall. The cell wall is constructed of two main parts called valves. These two parts fit together more or less as like halves of a petri dish. The living matter, the protoplasm, resides within the space between the valves. There are also bands of silica around the outside holding the two valves together. Much of the study of diatoms requires digesting or somehow destroying the living matter and then separating the valves. Pictures that you will see here are mostly individual separated valves. There are two main groups of, I might want to comment here on what you're seeing. If you look at the diatoms from one direction, you see the views that are on the left, the elongate diatom or the round diatom. If you look at them on the edge, you see the two halves of the diatom made up of the two valves. There are two main groups of diatoms. There are the centric diatoms, which are usually round and have a radial symmetry. And those are shown there at the bottom. And that is typical of what you would see in a centric diatom. The pennate diatoms, which are usually elongated, and what you have there at the top. The pennate diatoms may be shaped like a canoe, or they may be curved in various shapes. Some are S-shaped, and we will see some other shapes that come along. Here is a colony of living diatoms. Notice that they are not as green as grass. This is because diatoms use a different mixture of pigments to capture the sunlight. They use chlorophyll, but the chlorophyll is not the dominant pigment shown here. This picture shows diatoms that have been treated and mounted on a slide. Notice the range of sizes and shapes. Incidentally, the colors on this slide are not the result of the protoplasm, but are an optical effect in the microscope. But you see that some diatoms, there's one on the bottom there, which is very small. One over on the left and lower part is extremely small compared to the big one in the middle. You also see a centric diatom there tucked in that cluster at the bottom. This is a scanning electron microscope picture of a centric diatom. SEMs, or scanning electron microscopes, can resolve much finer details than a light microscope, but they're also harder to use. These are the halves of two different diatoms held together by the teeth on the one side. If these two cans represent the two valves of this diatom, they would be in a position like this in life while they'd be together. The living protoplasm would be on the inside of these cans. After the diatoms die, the valves separate, but adjacent valves remain together, stuck together by these teeth that we saw in the first one. These teeth hold one half of one diatom to another half of another diatom together, and the protoplasm is all gone. I want to point out, though, that only some centric diatoms do this. Many are shaped just like a petri dish. They're much flatter, and they are not joined together as these are. Here is a light microscope picture of a chain of the kind of diatoms we saw in the last few pictures. This right here, if you can see my cursor, is one diatom. Here is another diatom, and up here is a third diatom. In between the diatoms, the teeth that hold it together. So what we were seeing was one valve from this diatom and one valve from this diatom in that last picture. This is a scanning electron microscope picture of a typical pennate diatom. Notice its overall shape is more like a canoe. Also notice the striae of rows of holes that we find, if you can see my cursor here, going out from the center toward the edge. Running down through the middle of the valve near the center to near the ends is a black streak on this photograph, which is the rafe. The rafe is a slit which is through the cell wall. It's found on many species of diatom or pennate diatoms, but not all. There are pennate diatoms that do not have this slit at all. The living diatoms that have this rafe can generally move about in the sediment on various substrates and even on a microscope slide. In this way, they resemble animals. And for a long time, the early investigators back in the 19th century didn't know if diatoms were plant or animal. We actually don't treat them as either today. Here is another scanning electron microscope picture of a pennate diatom, and the rafe in this one is this short little thing right here. This is the genus Eunotia. Notice the striae, the rows of pores that cross the whole diatom. This is a rather long diatom, and it has a little short rafe like this at each end. Now, if you live in a glass house, how do you reproduce? This is looking at the edges of hypothetical diatoms. Diatoms produce mainly asexually. The living protoplasm expands within the two valves, moving them apart. Then within each valve, another one is formed. When completely formed, the two valves, now two completely separate diatoms, separate. Because the one valve is slightly smaller than the other, the lineage along the lines of the smaller valves becomes smaller and smaller. The average size of the population decreases. And obviously, this can't go on forever. Two diatoms come together, infuse their protoplasm, produce a cocoon-like structure, and then make one diatom the size of the first one in the series. This constitutes sex for a diatom. Then the whole asexual reproduction begins again. As a result, this kind of reproduction, the individuals in any population of the same species have a wide range of sizes, small ones, medium, and big ones. Here's a picture of a variety of diatoms living together in a body of water. Now, these have been treated and separated, so we're only looking at the valves. But you see that they are clear. They come in a variety of shapes. There are some centric diatoms, those big round things. There's a smaller round one up in the upper left. There are that big long thing on the left side is a diatom, which has been much bigger than the field of the scope. There are tiny little diatoms in there, skinny diatoms, diatoms that are flat on one side and rounded on the other. And this is typically what one finds. The colors that you see in the next pictures are created by tweaking the optics of the microscope. If you just look at diatoms through the microscope without doing any tweaking or any filters, this is mainly what you see. Here is a somewhat blurry picture of a common pennate diatom called a Nitium, the genus Nitium. Notice the rafe running down through the middle and coming to the center, and both of the ends twist out in opposite directions. Right here, you can see the rafe coming down and going out. the rafe from the other end is coming down here and going out this way. Then you have all these striae, these rows of pores making up the surface. Here is a bone-shaped diatom found in very soft waters. I doubt any of them are growing here in Iowa, but you go to some northern lakes and they are rather common. Here's another common Pennate Diatom found in springs and soft water bodies here in Iowa, as well as many, many other places around the world. Notice how it's curved. It's got this canoe type shape to it, but it's like the canoe ran into a tree and got bent. That's also typical of many diatoms. Here's a mount that kept the colonies of diatoms together. Many diatoms do form colonies, and you see the straight chains there, which is one species, and then there's the radiate colony, which is made of another species. Here's a colony of diatoms that float in the water. Each of these spokes is a separate diatom. This is a diatom. That's a diatom. That's a diatom. This specimen came from Clear Lake in the spring. At that time of the year, Clear Lake is not very clear. And essentially all of the cloudiness of Clear Lake in the springtime is formed by diatoms in the water. Here's one of my favorites. This is Eunocia serra. You notice that the one side is serrated like a serrated knife. That is where the name comes from. That is like I say, a favorite diatom of mine. But there are other species on there. You have another pennate down there just below it, and you have another one over there on the far left, which is called Frostula. This is called Epithemia, the genus Epithemia, and the rafe comes down and goes like this and back out. And you can see this on some other ones that I will show you here in a little bit. This is a Meridian Allen Smithii that I described as a new species named after my brother-in-law. Can you guess his name? Allen Smithii. One cannot name a diatom after yourself, so you pick friends or brother-in-laws or people you hold in high regard. Here's another one. The big one here is Frostula balzii, named after the diatomist who introduced me to diatoms many years ago. The other two diatoms here are also Frostula, but they're Frostula of a different species. Here is a common Pennate diatom. Here's one that's a little different. Suriella is the genus of this one, and it is found in waters that are pretty soft, but not as soft as some others. Now, most of the photosynthesis by diatoms is in the ocean. Oceans cover most of the planet, and that is where most of the diatoms are found. This is where the oxygen in that one breath in 4 or 5 that you breathe comes from. But a lot of diatoms live in places like this. This is Bare Meadows, an acid fen in central Pennsylvania, and it has a rather unique assemblage of diatoms. Diatoms are fussy where they live. Some like acid waters, some like alkaline waters, some like waters with little dissolved salt, some relish salt. So by knowing the kinds of diatoms living or once lived in a body of water, you can get a measure of the salt, the acidity, and many other physical and chemical parameters of that body of water that exists or once existed. This is a lake in Scotland, and it was very pure water, and the diatoms there were completely different than anything you'd find in Bare Meadows. And you also see how we're sampling there. The man on the left is holding a plankton net, You can throw the plankton net out and pull it back, or you can drag it behind a boat, and it filters the diatoms and other things that are in the water and deposits them in the bottom of a bottle at the bottom of the sampler. The fellow on the right is probably scraping diatoms off of a rock, and that is another place where we find them quite often. Here I am sampling water out of a pond using a turkey baster. I use the turkey baster to suck up a little bit of the water and the diatoms in it, and then I place that into a plastic bottle. I have a little box that I carry around my sampling material and samples, and here is a closer view. You see the bottles, tops of the plastic bottles I put the samples in. You see the turkey baster, you see the droppers on the side, even a toothbrush over there, and also a GPS so that I can find where I took the samples and record that. And of course, I record all this stuff then in a notebook. Another sampling device is my canoe. I can go out into a lake or a big pond with the canoe and sample out there, and it comes in handy sometimes. Copper dredge, which I sometimes throw out into a stream or a lake and then retrieve it using the rope. It works very well. Getting the sample is sometimes easier than getting to where you want to sample. I once crawled into a wetland in North Carolina through rhododendron and swamp roses. When I emerged, I was covered in scratches and bleeding all over. Enough hero talk. This is more typical scene of diatomists at work. Actually, this was a lunch break on a field trip. You might wonder what commercial uses the diatoms are used for. Diatomite is the term, the rock the name we assigned to the rock, which is really a collection of fossil diatoms. Diatomaceous earth is another term which is often used. Diatomaceous earth is sold to be used as insecticides, and I have a friend who uses it as insecticides. She also uses it to condition the soil in her garden or in flower pots. The diatoms work against insects by scratching their exoskeleton, and they dehydrate and die. Diatomite or diatom material is used as a polishing agent and abrasives, and once was used as toothpaste, but that's not a good idea because the diatoms are harder than the enamel of your teeth. So if you brush your teeth too much with diatoms, you'll wear away your teeth. It's also used as a filtering agent. The last beer you drank, if you drink beer, may have been filtered through diatomite. The cooking oil that you used in frying some eggs this morning was probably filtered through cooking oil. And sometimes waste materials are filtered through diatomite. I understand that one time, maybe still today, John Deere uses diatomite to filter some of its waste material that goes into the river. It's also once used as dynamite. I don't know what they make dynamite out of today, but the first dynamite was soaking up nitroglycerin into diatomite. Is dynamite and diatomite two different words, and I don't know if they're connected or not. This rock, the diatomite, is mined like many others. It's light in weight and quite soft, and here you see some of its uses. Now we come to Hartman Reserve. There are diatoms in that stream. I took some samples from near the K. Ramanan Bridge here in the park, or reserve. A few days after a rain, the stream flow slowed down and the diatoms grow on the surface of the mud in the rocks. That is where I got the following diatoms. I did not sample the Cedar River, but had I done so, I would have had many, many more types, and that river is full of diatoms. Sometimes billions of diatoms float by every second. Here is one of the ones from Hartman. It is genus Calaneus. You can see the striae on the sides. You can see the rafe running from near the center out toward the ends. In this one, you can also see some other diatoms over on the left. Here's my cursor. There is the rafe coming in, making this little bit of a bip, and then going out the other way. And that is characteristic of epithemia. Epithemia, I might point out, are found, some of the same species found here in Iowa and some of the wetlands that are a little bit more salty. And they're also found in the upper part of Mobile Bay, same species. This is Criticula, rather common species that you find, and it's here in Hartman Reserve. Here is Pinularia. Pinularia can be some really big diatoms. And you can compare that diatom in the middle to the one down in the lower part, which is much, much tinier. And also the one up above, which is shown on the side, and that's much tinier too. Here's another epithemia with a little rafe coming in and doing a little dimple there in the center. And this concludes this presentation. I hope that you have found it interesting and informative. Diatoms are part of Hartman Reserve and the rest of the world around us that we don't normally see. However, with a minimum amount of equipment and a microscope, you too can collect diatoms and view them at home or in the classroom. For your information, I have assembled a guidebook to study diatoms in the classroom, and it works at your home too. You may contact me through the Hartman Reserve, and I can see that you get a copy. Call Hartman Reserve and talk to Connie. Thank you for your attention, and goodbye.
Thank you, Lynn. That was really interesting, and I learned quite a bit each time I've heard you do it. And thank you for tuning in, everybody, and your support of Black Hawk County Conservation. We'll see you next month at same bat channel, same bat time. Actually, it's about, I believe, February 14th at 2 p.m.